Hi friends, in the last video it was about the acetic acid production. In this video it is about the third acid we are going to discuss, which is the production of gluconic acid. Gluconic acid, it is an organic compound and with molecular formula C6H12O7. The structural formula when it condenses is given, and it forms a gluconate ion in aqueous solution at neutral pH. The salts of gluconic acid are known as gluconates. Gluconic acid and its salts and esters occur naturally in fruit, honey, and wine as they arise from the oxidation of glucose. A bit of history. In 1911, Alsberg explained the production of gluconic acid, in which the fermenting microbe was Pseudomonas, and in 1928 its production using the fungus Penicillium luteum by surface fermentation process was employed for the first time. At present, it is commercially produced by using the fungus Aspergillus niger or the bacterium Acetobacter suboxidans. Mainly used process is submerged fermentation. In this process, gluconic acid, sodium and calcium gluconate and glucose oxidase are produced. Apart from these two microbes, others can also be used but not for commercial production. Such organisms are fungi, endomycosis, Pullularia, Penicillium, Gonadotropis, and Scopulariosis, and the bacteria Pseudomonas and Vibrio. Gluconic acid is produced from glucose, which is catalyzed by glucose oxidase. Gluconolactone is formed at the end of the first step and undergoes hydrolysis, which is either spontaneous or enzymatic, to finally produce gluconic acid. During the first reaction, the hydrogen from FADH2 is transferred to oxygen, leading to the formation of H2O2. To prevent its antimicrobial activity, it is immediately split into water and oxygen by catalase enzyme. Fermentation process of gluconic acid. For industrial production, the fungus and the bacterium are employed. In the former process, Aspergillus niger, and in the later process, Acetobacter suboxidans are used. A niger employs glucose oxidase involving agent like FAD and lactonase in the presence of oxygen, resulting in the formation of gluconic acid. On the other hand, gluconobacter employs glucose dehydrogenase with the coenzyme pyrroloquinoline quinone, which is in short form PQQ, and lactonase, which help in dissipating the hydrogen peroxide. Fungal fermentation. Submerged fermentation process is used for fungal fermentation. Aspergillus niger is the microbe. Either sporulated culture or spores germinated in the seed tank is used as the inoculum. Each has its own advantages, like for example, use of spore inoculum directly avoids the cost of installation and operation of the seed tank, while the use of germinated spores reduces the operating cycles for the main fermenter. So this is the diagrammatic representation of what we discussed earlier, and here we can see glucose is converted to alpha gluconolactone and then finally to gluconic acid. In the presence of glucose dehydrogenase, and here in the presence of catalyzed by lactonase. So this is the formation of gluconic acid from glucose by Acetobacter suboxidans. Here in this figure we can see here an A is there, and this is figure B, And in this figure A and B, it is the gluconic acid formation by enzymatic reactions by using G suboxidants, and in this B figure it is the gluconic acid formation by Aspergillus niger. Glucose is used as a solution or crystalline glucose or in the form of a syrup prepared from starch or crude starchy material which are treated with amylase or amyloglucosidase. So this is the composition of gluconic acid production medium. Here we can see the substrates or the chemicals which are used, and the quantity to be used are written here, or the percentage also. This is the medium used for the gluconic acid production. Turbido mixture or cavitator is used for vigorous agitation to maintain the maximum concentration of dissolved oxygen in the form of solution. In the manufacture of calcium or sodium gluconate, optimum temperature is between 28 to 30 degrees Celsius and the pH is 6.5 along with vigorous agitation and aeration. Recovery and harvest. After the fermentation, fungal mycelium is separated by filtration. The separated mycelium is then used for the recovery of glucose. On the other hand, the filtrate is used for the recovery of calcium or sodium gluconate. Production of calcium gluconate. The steps are: the filtrate is heated with the excess of calcium hydroxide and the resulting product is decolorized with the activated carbon and filtered. The compound is then crystallized by cooling at a temperature below 20 degrees Celsius and the seeding with calcium gluconate crystals. The liquid which is left over, which is about 10 to 15%, is used for the production of second crop of calcium gluconate through heating, followed by the treatment with the carbon filtration and chilling. Production of sodium gluconate. The steps involved in production of sodium gluconate is: the filtrate is concentrated to about 40 to 45%, and then sodium hydroxide is added which crystallizes the sodium gluconate and also maintains the pH at 7.5. The sodium gluconate formed is then drum dried. Production of pure gluconic acid. Here calcium gluconate is employed and the steps are: first one, the calcium is precipitated by the addition of sulfuric acid and the calcium sulfate formed is separated by filtration. The filtrate is decolorized using activated charcoal. The acid solution is then concentrated to 50% acid strength. The product formed is a mixture of gluconic acid and lactones. The concentrated is then treated with a temperature ranging from 0 degrees Celsius to 30 degrees Celsius. The crystals that separate at this temperature consist of pure gluconic acid. When the concentrate is treated with a temperature ranging from 30 to 70 degrees Celsius, crystals of lactones are separated. Why lactone crystals are separated when the concentrate is treated with a 70 degrees Celsius and above temperature? Bacterial fermentation. First we told about fungal fermentation. Now it is bacterial fermentation, in which submerged fermentation is used. The acetobacter suboxidants and gluconobacter suboxidants are used. In the pure culture of the bacterium, it is raised by repeated subculturing process and is used in the fermentative production of gluconic acid and is produced either in the form of calcium gluconate or sodium gluconate. Glucose is used as a substratum in the fermentation. If calcium gluconate is to be produced, 13 to 15% glucose is added as a substrate due to low solubility of calcium gluconate. Higher calcium gluconate levels are formed if glucose is used above 13% levels, which would spontaneously crystallize as calcium gluconate and makes purification difficult. 28 to 30% of glucose concentration should be used if sodium gluconate is to be produced. The fermentation is carried out at a temperature of 28 to 30 degrees Celsius and the pH is between like 4.5 to 6.5 with a very high aeration rate of 1 to 1.5 VVM. VVM is the volume of air under standard condition per volume of liquid per minute. The gluconic acid yield can be substantially increased, that is 90 to 95%, by increasing the solubility of oxygen, which is achieved by raising the pressure in the system. Now the uses or applications of gluconic acid, it is used in the manufacture of metal, leather and food, and sodium gluconate for sequestering agent in many detergents, and calcium gluconate is used in medicine. Gluconolactone used as baking powder and as an additive. This is all about the production of industrial production of gluconic acid. Hope you all understood. Thank you.